And we're very happy because now this is the final composition. Now, according to the FEA models and according to literature, the maximal stress in a tibia or in a femur is around 20 megapascal, so which is here. And our fracture point is at 70, which means that we can sustain a 3.5 body weight of the sheep, in this particular case, before this material will crack. So we are kind of confident that the sheep will walk after we take away that 5 centimeters of femur or of the tibia. So that takes account of months of work on the biomaterial development. As I said before, there will never be the good regeneration of the bone if we don't have the right biology on board. And the right biology is driven by the right factors. And so mesenchymal stem cells are the players, but also the mesenchymal stem cells have to be recruited and retained in the scaffold. They have to be forced or stimulated to proliferate and eventually to differentiate in the type of bone cells that you need. So we just developed a lot of different delivery system, and this is what we're good at UTH. This is the work of my group. So we develop all these silicon structures. They're porous. They're very cheap to make. They're very reliable in terms of size and shape, reproducibility. We have different size and shapes. We have different porosities. This is all in the same scale. So the advantage of this is that these guys have pores all the way through the structure, and you can load them and pack them with nanoparticles, growth factors, biomolecules, and once they're inside, I'll show you later, they're protected from degradation. So different sizes of pores according to what you need to load inside, so that they can accommodate from a very tiny molecule, 2 nanometers, to a big liposome, like 150 nanometers. We also develop a lot of different coating strategies, capping strategies to match the requirements of these time staggered release of the molecules. So now we managed to have releases in terms of hours, in days, or in weeks, according to the different recipes and the different structures. We tested all the different structures, all the different growth factors, in order to identify what was the cocktail, the best cocktail of factors that could promote the cell proliferation. So all this work now pointed out at this final composition, and that's what we're using at the moment. As I said before, the silicon is a great way to deliver biomolecules because you can control the release, but also because once they are loaded inside, they do not degrade. So this is just BSA, it's a model protein, and these are all the degradation byproducts if you leave it as is, without even adding trypsin, just by leaving it as is for 7 and 14 days. Now, if you load it inside the silicon, you don't see those degradation bands anymore, which means that the protein is stabilized. Also, you can control the internalization of target cells so that you can decide if you want or not them to internalize your particles. They completely degrade, they're completely benign. And actually, the good thing is when you combine PLGA and nanoporous silicon enclosures, NSCs, the degradation byproducts respect the neutral pH, which is very important because most of the polymers that are used in the bone tissue engineering, such as PMMA, they say they're biocompatible, but they create a hell of inflammation because they are like the byproducts are very acidic. So that's something that we wanted to be very careful about. So now the amphiphilic peptides, a quick thing. So you need the cells, you need the right materials, and you need the right environment. And these amphiphilic peptides in the right composition, they promote the deposition of minerals. And the good thing is that the deposition of minerals is also very close to the ratios that are typical of natural bone. So natural bone is 1.657, this is 1.65, so very, very close. And you see the way they assemble, they created this dense network of fibers, which resembles very closely the extracellular magic, so cells love this thing. Then let's go to the cells. The cells have to do a bunch of stuff. That they have to stimulate the angiogenesis, They have to recruit other mesenchymal stem cells. They have to proliferate and differentiate, and so on and so forth. We follow two approaches. We use either the cells that can be derived from the bone marrow, so from just a bone marrow aspirate with a few of a little bit of cleaning with fax, or we isolated this new class of cells, the compact bone. So by taking the bone itself, crushing it a little bit, and doing some other magic, which I can't say, but anyways, we isolate these compact bones, and the amazing thing of these compact bone cells is that they grow 40 times faster and better than the normal bone marrow-derived cells. So these cells are, what does it mean?
It means
5 minutes. Oh my God!
But if you want to go longer, there could be less questions.
Okay.
Talking to you.
Okay, I'll go faster. So, anyways, they differentiate in all the different lineages, and we also tested the way they assemble on the scaffolds. Now, I'm not sure that you can appreciate that this is like a Z-stack throughout 200 microns of the scaffold. And you see in red the particles, like the delivery system, in green the cells. And you see that they are dispersed throughout. So it's very uniform the way they grow inside the scaffolds, which is, again, it was not a piece of cake to get. And you see, again, in confocal analysis that they never co-localize. Again, you don't want the particles to get internalized by the cells because the particles release growth factors. Growth factors are active outside. If they get internalized, they're useless. And so, again, this is another reconstruction, and you see that they're never together, even though they grow throughout the thickness of the scaffold. Of course, another very important thing, the scaffolds need to degrade eventually, as we said, and they have to degrade in the arch of 6 months, because if they degrade too fast, then the bone didn't really develop at the point in which it can sustain all the loads and the forces that act on it. So we had to do a lot of different recipes, and we're now kind of happy because the bone formation starts officially at 1 month. We try now to anticipate it a little bit better, and according to the different formulations, that's when basically the degradation starts. And a long time, you see that the scaffolds became more and more porous. You have to imagine now in these pores the bone tissue that has been regenerated that is infiltrating and filling the voids and the gaps. And again, we always keep an eye on what happens in terms of pH, because something as simple as pH can mess up the whole experiment, and we're happy because we keep it around 7.4. No matter what is the composition. We also analyze all the byproducts. The mathematics, let's keep it super fast. Now, we're doing now a lot of mechanical testing. We work with a lab that has incredible abilities in testing implants and scaffolds. So they developed a lot of tests. Now, this that seems like a big bone, this is the bone of a rat. So it's something like this big. But they managed to develop a test so that we can understand what are the mechanical properties of the materials when we implant it in the bone of a rat. So without touching the sheep, which is of course more expensive and more a little bit trickier to deal with. So they have all these FEA models that they developed. They can merge the micro CT with the mechanical testing so that they see the displacement, the tilting, and this eventually got to the miracle, the dead sheep walking. So basically, even before, that's how we called it. It's a gait simulation. So basically, this is the leg of a sheep, of a real sheep. So we take the whole leg, and they rebuilt it on this machine, studying the different ways the sheep walks. Because we don't want to get to the point of doing surgery on a sheep, and after 1 week, the sheep stands up and breaks the bone. We don't want that. So we recreated this system, and we animated the sheep leg. So now, even if it might be kind of gross, but it's very useful for the surgical team, because we can also do surgery. These have muscles, tendons, everything in place. It's just explanted. We can put the implants here and check if the mechanical properties will be solid enough to sustain all the loads that can be applied in all the movements that are usually performed by a ship while it moves.